Metastatic melanoma. It's still a really tough diagnosis, isn't it?
It absolutely is.
I mean, we've seen some amazing progress, especially with immunotherapies.
Definitely. Things like CHEK2 inhibitors have been game changers for some.
But still, resistance is a massive issue, and that prognosis, well, it can still be quite grim for many. The 5-year survival rate, you mentioned, it's around 37%.
Yeah, roughly. And that number really highlights why we can't stop looking for answers.
Yeah.
We focus a lot on the genetics, which is obviously critical.
Sure.
But it's clearly not the whole picture.
Right.
We need to explore, well, everything else that might be making these cells so tough.
Exactly. And that's why we're doing this deep dive today. You sent over this really interesting research paper that looks at one of those, let's say, less talked about factors. So our mission is basically to unpack this study together, see what it tells us about why melanoma can be so aggressive.
Right. And this paper zeroes in on the tumor's own little world, its microenvironment. Specifically, how low oxygen levels hypoxia actually change the building blocks on the cell surface.
The building blocks.
Yeah, these complex sugar chains called glycans. They're all over the cell surface. The paper explores how hypoxia messes with these glycans and how that links up with another player, a protein called Galectin-8.
Okay.
And the idea is that this whole interaction, this chain of events, might be really important for the melanoma's nasty characteristics.
Okay. Low oxygen changing cell surface sugars. That already sounds, well, pretty fundamental, but not something you hear about every day. So the plan for today, we'll quickly recap the challenge, then focus on hypoxia in the tumor.
Yep.
Then we'll get into the key players, these glycans, an enzyme, GCNT2, Galectin-8. We'll look at how the paper shows they interact.
And connect the dots.
Exactly. Connect the dots and see if this gives us any new ideas for, maybe new biomarkers or even treatments down the line.
Sounds like a plan. Let's jump in.
Okay, so setting the stage, metastatic melanoma. We know it's hard to treat. Immune therapies work wonders sometimes, but then resistance kicks in for too many patients.
It's a constant battle. That 37% survival figure really hammers home the need for fresh approaches.
And like you said, a lot of focus has been on genetic mutations, the immune response, but this paper points elsewhere.
Right. It pushes us to look beyond the DNA at things like, well, how the cell actually builds itself, especially its surface, and how it adapts to the really stressful conditions inside a tumor. Glycosylation is one of those areas.
Say that again?
Glycosylation. It's basically the process of adding sugar chains, the glycans, onto proteins and fats.
Ah, right. And these aren't just like decorations.
Oh, not at all. They're super important, especially on the cell surface. They affect how cells talk to each other, how the immune system recognizes them.
Yeah.
All sorts of critical stuff.
Got it. So let's talk about that stressful environment. Hypoxia, low oxygen. How low are we talking in these melanoma tumors?
Well, the paper mentions it can get really low, down to like 1.5% oxygen sometimes, which is pretty extreme compared to normal tissues.
And low oxygen is generally bad news in cancer, right? It's linked to those tumor-initiating cells.
Exactly, TECs. These are sort of the stem cells of the tumor, really resilient, good at dodging treatments, evading immune attacks, and basically driving relapse. Hypoxia is known to help create these cells.
Okay.
But what's cool about this source material is its specific question. It's not just saying hypoxia is bad. It's asking, how does hypoxia specifically change the glycome?
The glycome, so like the cell's entire set of sugars.
Precisely. Does the low oxygen actually change the sugar landscape on the cell surface? That's a much less studied angle compared to, say, its effect on genes like HIF1A.
Okay, so how did they figure out if the sugar landscape changes?
They started with RNA sequencing, looking at which genes are switched on or off in melanoma cells under normal oxygen versus low oxygen.
Like a snapshot of the cell's active instruction manual.
Exactly. And the snapshot showed pretty clearly that hypoxia significantly messes with the activity of a whole bunch of genes involved in making these glycans.
Wow. So the low oxygen is basically telling the cell, okay, time to build your sugar coat differently. Any specific instructions that stood out?
Yes, a couple really caught their eye. One gene, GCNT2, it makes an enzyme that adds branches to these sugar chains. Think of it like building side roads off a main sugar highway.
Okay, branching enzyme.
Yeah, GCNT2 activity went down under hypoxia. It was downregulated. But another gene, LGALS8, which codes for that Galectin-8 protein we mentioned.
The one that binds to sugars.
Right. Its activity went up, upregulated.
Huh. So less branching enzyme, more sugar-binding protein. That sounds coordinated.
It really does. And they didn't just rely on the RNA-seq. They double-checked these specific gene changes using another method, RT-qPCR, which confirmed GCNT2 down, LGALS8 up.
Okay, so the gene instructions change, but does it actually show up on the cell surface? Because that's where it matters, right?
That's the key question. So they use flow cytometry. This lets you analyze the surface of thousands of cells really quickly.
Right.
And bingo! Under hypoxia, the cells showed more Galectin-8 protein physically present on their surface. And interestingly, they also showed an increase in certain glycan structures called I-linear poly-LacNAcs.
Okay, hold on. I-linear poly-LacNAcs. Let's break that down. What are poly-LacNAcs?
Okay, so poly-LacNAcs are essentially chains made of repeating sugar units. They get added onto bigger glycan structures, usually N-glycans, which are attached to proteins. Think of them as extensions or modifications on the main sugar tree.
Got it. Repeating sugar units. And I-linear versus, what was the other one? I-branched.
Exactly. It's about how those repeating units are linked together. I-linear means they form a straight chain. I-branched means, well, there are branches coming off that main chain. Same building blocks, different architecture.
Straight chain versus branch chain. Okay.
Yep.
Did the cells normally make both?
Yeah, but the paper showed different cell lines had different preferences, even in normal oxygen. They used mass spectrometry for this, basically weighing tiny fragments of the glycans to deduce their structure. One cell line favored the linear type, another favored the branch type.
But the kicker is what happened under hypoxia.
Right. The mass spec data was pretty clear. When oxygen levels dropped, both cell lines shifted towards having more of the I-linear structures relative to the I-branched ones.
Ah, so hypoxia pushes the cells to make more of the straight chain version.
Yep. There was a definite increase in the ratio of linear to branched.
And that ties back perfectly to that GCNT2 enzyme, right?
Yeah.
The one that makes the branches and gets turned down by hypoxia.
Exactly. GCNT2 is the I-branching enzyme.
Yeah.
So if you reduce its activity, it makes perfect sense that you'd get fewer branch structures and, relatively speaking, more of the linear ones that don't need it.
It's like the cell's losing the tool to build the side road, so it just keeps building the main highway straight.
That's a good way to put it. It's a very specific structural change on the cell surface, driven by the low oxygen environment.
So this specific sugar change, more linear, less branched, does it actually mean anything for patients? Does it correlate with how they do?
Great question. They looked into that. They analyzed patient tumor samples and measured the levels of that GCNT2 enzyme, and they found something pretty striking. Lower levels of GCNT2 expression in the tumors correlated with significantly shorter survival times for patients who ultimately died from melanoma.
Wow, okay, so having less of that branching enzyme seems to be linked to a worse outcome. That could be really important.
It really could. It suggests GCNT2 levels might be a potential prognostic marker, something you could measure to get a sense of how aggressive the disease might be.
Did they try to prove that link more directly, like manipulate GCNT2 in the lab?
They did. They experimentally lowered GCNT2 levels in melanoma cells, kind of mimicking the hypoxic effect. And when they did that,
What happened?
they saw an increase in markers associated with those aggressive tumor-initiating cells like NGFR, which you mentioned earlier.
So reducing the branching enzyme directly made the cells look more like those tough teaks?
Exactly. It provides a direct experimental link. Less GCNT2 leads to fewer branched sugars, which seems to promote these aggressive therapy-resistant cell traits.
Okay. Let's bring Galectin-8 back in. We know hypoxia increases it on the cell surface. Is it also higher in patients generally?
Yes, they checked that too. They measured Galectin-8 levels in blood serum from melanoma patients versus healthy controls using ELISA tests. Significantly higher levels in the melanoma patients.
So Galectin-8 could potentially be a biomarker too, maybe something you could detect in a blood test.
Potentially, yeah. Elevated serum Galectin seems associated with the disease.
And how does this Galectin-8 interact with the sugars? Does it care about the branching?
It definitely seems to. They did binding experiments using purified Galectin-8 protein. They found it preferentially stuck to the melanoma cells that had more of those I-linear straight-chain poly-LacNAcs.
Ah, okay. So hypoxia drives up the linear sugars, and it drives up Galectin-8. And Galectin-8 specifically likes to bind to those linear sugars. It's all fitting together.
It really clicks into place, doesn't it?
So what exactly is the Galectin-8 binding to on the cell surface? Is there a specific docking station?
Good question. They went fishing for its binding partners using techniques like affinity chromatography and mass spec.
And they caught something.
They did. A major hit was a protein called CD44.
CD44, I know that name. It's heavily implicated in cancer progression, metastasis, right?
That's the one. A well-known player in cell adhesion, migration, and signaling, often linked to more aggressive cancers. So Galectin-8 seems to be latching onto CD44, likely where these I-linear glycans are present.
Okay, so under low oxygen, more linear glycans, more Gal-8, and Gal-8 binds CD44. What happens then? Does that binding send a signal?
That seems to be the critical output. The paper provides evidence that this binding event triggers intracellular signaling. They showed this by adding external Galectin-8 protein to the melanoma cells.
Like mimicking it binding from the outside.
Exactly. And when they did that, the expression of that teeter marker, NGFR, went up inside the cells.
Whoa. So binding on the outside ramps up aggression markers on the inside.
It strongly suggests a signaling cascade. Binding signal, more T-like state.
Did they try the reverse? Reducing Galectin-8?
Yep. They used gene silencing techniques to knock down Galectin-8 expression within the melanoma cells, and NGFR expression went down. Less Galectin-8, less NGFR. It reinforces the connection.
Okay, this is compelling in cell cultures, but does it hold up in a real living system? Does Gal-8 actually help tumors grow?
That's the million-dollar question, right? And crucially, yes, they addressed that. They injected melanoma cells into mice, the standard in vivo tumor formation assay. Some cells had normal Galectin-8, others had Galectin-8 knocked down.
And the results?
The cells with less Galectin-8 were significantly worse at forming tumors in the mice. Their growth was inhibited.
Wow. So Galectin-8 isn't just hanging out on the surface, it's actively helping the melanoma establish and grow in a living organism.
That's what the data strongly suggests. It seems to be a functional contributor to tumorigenicity.
Okay, let's try and piece this whole story together then. Based on this source material, you've got hypoxia, the low oxygen in the tumor.
Right.
That flicks some switches. It turns down the GCNT2 enzyme, meaning fewer branched sugars, and turns up the Galectin-8 protein.
Yep.
This leads to more of those straight-chain I-linear glycans on the surface. Galectin-8 loves these and binds to them, particularly on the CD44 protein. And that binding event sends a signal inside the cell, boosting markers like NGFR, pushing the cell towards a more aggressive tumor-initiating state and ultimately helping the tumor grow.
You've got it. That's the pathway they've mapped out. It's a really elegant example of how the environment low oxygen can trigger these specific molecular changes on the cell surface that alter the cell's fundamental behavior.
And it's so interesting because it's not necessarily about new DNA mutations, right? It's about the cell adapting its existing machinery, its glycosylation, in response to stress.
Exactly. It highlights this whole other layer of regulation in cancer involving the microenvironment and these surface glycans that we're probably still only scratching the surface of.
Which is why digging into sources like this is so valuable. It potentially opens up completely new ways to think about fighting melanoma.
Absolutely. If low GCNT2 means worse prognosis, maybe that's a marker. If high Gala-8 is found in patient blood, maybe that's a marker for detection or monitoring.
And thinking therapeutically. If Galectin-8 binding to CD44 via these specific glycans is driving tumor growth,
then maybe you could block that interaction, target Galectin-8 itself, maybe develop antibodies or inhibitors, or perhaps even go after those specific I-linear glycan structures.
could be totally new targets.
Yeah.
Maybe things that could work alongside immunotherapies, hitting the cancer from a different angle.
That's the hope. Finding these novel pathways gives us new potential vulnerabilities to exploit. It really underscores the importance of looking, beyond the usual suspects sometimes.
It really does. It makes you wonder. We focused on oxygen here, but tumors have all sorts of weird environmental conditions, low pH, nutrient stress.
All sorts of things.
Thinking about how hypoxia reshapes the cell surface like this, what other environmental cues might be sending signals through these kinds of glycan pathways, hiding mechanisms we haven't even considered yet?